Convergent evolution is the independent acquisition of similar traits in distantly related species. There are many examples of convergent evolution, from the development of wings in bats and birds to the shape and color of dolphins and sharks. While biologists have long been interested in this phenomenon, we have not been able to investigate the genetic basis of convergent evolution until very recently. Thus, there are many unanswered questions in this field. Do these traits arise through mutations in the coding sequence of genes that change how they function, or do they come from mutations in regulatory regions that alter when and where these genes are expressed? Are the same types of mutations used by different species to develop these traits, or do different species evolve similar traits through different mutations in different genes? Unfortunately, many examples of convergent evolution are in complex traits that are controlled by multiple genes, making these questions difficult to answer. Hello, I'm Matthew Treaster, a graduate student in the Department of Genetics at the University of Georgia. I researched the molecular mechanisms of evolution using Threespine stickleback fish, a well-established model for studying how a variety of developmental processes evolve. Like humans and other mammals, Threespine stickleback have an XY sex chromosome system, where a sex determination gene on the Y chromosome causes individuals with that Y to become males and those without it to become females. However, the stickleback X and Y have evolved completely independently of those in mammals. In fact, we know that many different species have evolved genetic sex determination using sex chromosomes to determine which individuals are male or female. This genetically simple single-gene mechanism of sex determination provides a unique opportunity to study convergent evolution in fine detail. Using the gene editing tool CRISPR-Cas9, I have demonstrated that the gene AMHY has evolved the function of sex determination in Threespine stickleback, as XY fish without AMHY develop ovaries rather than testes. AMHY is a second copy of the gene anti-Mullerian hormone that has been duplicated onto the stickleback Y chromosome. Amazingly, this same gene has been duplicated in multiple vertebrate species and convergently evolved this same function. However, how AMH repeatedly evolves function in sex determination is not yet known. My future research will focus on this mechanism by which AMHY controls sex determination, as well as how it evolved this function. By characterizing the expression pattern of AMHY, I will determine if newly evolved regulatory elements contribute to its novel function in sex determination. Then, I will identify downstream genes and pathways targeted by AMHY that allow it to control this process. My findings will not only enhance our understanding of the critical developmental process of sex determination, it will also facilitate comparative research investigating the evolution of AMHY and other sex determination genes across a variety of vertebrate species, answering those fundamental questions about how convergent evolution occurs at a molecular level. Thank you for watching, and thank you to the ARCS Foundation for considering me for this award.